This is a very short lecture on recombination. The objectives of this lecture are to define recombination and illustrate the modes of recombination among sexually and asexually reproducing organisms. At the end of this lecture, you should be able to list the advantages of recombination, define and sketch a bacteriophage, and compare and contrast conjugation, transduction, and transformation. Recombination occurs in both eukaryotic and prokaryotic cells. And what recombination does is it produces offspring that have new genetic combinations, which are called recombinants. Recombinant organisms are naturally produced in eukaryotes through sexual reproduction. However, in prokaryotes, recombinant organisms are produced through three different processes. One is conjugation, one is transduction, and the other is transformation. In sexually reproducing organisms, recombination occurs through crossing over, which is physical exchange between molecules. So if you remember when we were talking about cell division and meiosis and mitosis, we talked about crossing over occurring between the homologous chromosomes. So each generation of sexually reproducing organisms has a new combination of the parental genome, and this recombination is based on Mendel's law. This is a schematic of crossing over, where you have genes that are transferred between the two chromosomes to make a recombinant chromosome. There are several advantages of recombination, and the two strongest advantages are to increase the robustness and adaptability of offspring. And there's this concept of hybrid vigor or heteresis. And this is the concept where mixed individuals are stronger than purebred organisms. Recombination occurs during meiosis when we have those duplicated homologous chromosomes that line up and recombine through crossing over. Those recombined chromosomes are then randomly assorted into gametes so that each gamete contains a set of the recombined parental chromosomes. So the gametes have the chromosomes from the parent, but they've recombined in such a way that the gamete have a unique set of chromosomes. This is a schematic of this event where you have your homologous chromosomes that cross over. So you have new genes in these chromosomes. These recombined chromosomes or recombinants are then randomly inserted and form your gamete. So each gamete has a unique set of the parental chromosomes. Now, in asexual reproduction, recombination occurs via three different processes. There's conjugation, transduction, and transformation. And we are going to keep these very simple. So in conjugation, you have F plus cells and F minus cells. These cells must physically come together in order to conjugate. So these cells actually form a bridge between one another, and the F positive cells will transfer their genetic material to the F negative cells. Thus, the F negative cells become F positive after conjugation is complete. In transduction, you have bacteriophages. Bacteriophages are viruses that infect bacterial cells, and these bacteriophages insert genetic information so that the cell obtains new genetic information. In transformation, you get DNA inserted into a bacterial cell. And this DNA can be DNA that was left over from a degraded cell, or it can be plasmid DNA. And we're going to discuss a lot more about plasmids in a later lecture. So in conjugation, we have a schematic here where you have an F minus bacterial cell and an F plus bacterial cell. They bridge together, or it's their bacterial form of mating. And you have the F factor DNA that gets inserted into the F minus cell. So now both cells become F positive cells. In transduction, we have our bacteriophage, or a virus that infects bacteria. The bacteriophage goes in, sits on the bacterial cell, and that phage is almost like a needle and inserts some of its DNA into the bacterial cell. That the DNA that it inserts into the bacterial cell can then become part of the genomic DNA of that cell. This is a schematic of a typical bacteriophage, and a bacteriophage is just like any other virus where you have your capsid and you have your DNA or RNA genomic material. The difference between the bacteriophage, like I said, is this contractile sheath that acts like a syringe to inject genomic material into a into a bacterial cell. Now, in transformation, you get DNA fragments. These DNA fragments can either be pieces of plasmid DNA, they could be pieces of genomic DNA from a cell that's recently lysed. These fragments of DNA can then be taken up by a recipient cell, and then they can be integrated into the chromosome of that cell. So, in summary, we have recombination in sexually reproducing organisms via crossing over and random chromosomal combinations among the gametes. In asexual reproducing organisms, you get recombination either through conjugation, transduction, or transformation. And it's our cartoon, and due to recombination events during replication and cell division and mitosis and meiosis, we are all mutants. We all have a varied form of our parental chromosome. So that's what makes each and every one of us a unique individual.